This video demonstrates a method for inducing cataracts using ultraviolet radiation in the rat. The molecular mechanisms involved in cataract development are subsequently studied using quantitative real-time PCR and immunohistochemistry. First, one rat's eye is exposed to ultraviolet radiation to induce cataract formation. The other eye serves as a control. Next, the lenses are dissected and processed to obtain RNA to assess caspase-3 expression levels by quantitative real-time PCR, or stained using an antibody against the cleaved caspase-3 to assess the expression and localization of active protein. Ultimately, the results from these assays confirm an increase in caspase-3 mRNA and expression of the active protein in the nucleus of the lens cells following exposure to UVR.
The main advantage of this protocol over existing method is that it uses a unique narrowband UVR source and universal red restrainer, allowing us to safely and precisely expose animal eyes with well-defined UV spectrum and dosage. Moreover, we applied modern biological techniques, quantitative RT-PCR and immunohistochemistry to study apoptosis in the lens.
Though this method can provide information about molecular mechanisms of ultraviolet radiation-induced cataract, it can also be applied to other experimental models such as ultraviolet radiation-induced skin cancers, rat behavioral neuroscience, and experimental eye research with focus on apoptosis.